Hello, this is a leukocyte lecture for A&P2. My name is Gina Piscatelli at Madison Area Technical College. Leukocytes are referred to commonly as white blood cells. In this picture, I think it's been color enhanced. Most of the time, everything looks kind of a generic gray color, but they certainly don't look red like red blood cells due to the iron in hemoglobin. So leukocytes aren't as prevalent in blood as erythrocytes are. A normal count is less than 1% of blood volume, and 5,000 to 10,000 cells per microliter. Their general function is defense and immunity, protection against infection. And there's two categories of white blood cells. Those that are called granulocytes and those that are agranulocytes. And this refers to the visible granules that you can see in the cytoplasm of some cells. For example, the cell labeled E in the diagram, you can see these bright pink circles in the cytoplasm. Those are called specific granules. Even neutrophils have them, the cell labeled N, and basophils have those. But the cells that don't are called agranulocytes, and they include lymphocytes and monocytes. So there's a diversity of white blood cells. They vary in size, shape, even number. They're all bigger than red blood cells, though, and they all have an obvious nucleus. They also retain all their organelles so they can produce proteins because they have ribosomes and DNA in a nucleus. They all contain certain inclusions or organelles called lysosomes. And those are organelles that contain the enzyme lysozyme, which can be released to degrade extracellular material. But the granulocytes have more granules called specific granules. And these granules or organelles contain different enzymes called defensins, and they're specific depending on which granulocyte you're talking about, or unique, I should say. So the defensins vary from cell type to cell type. Like an eosinophil has certain enzymes and a neutrophil has different enzymes. And so those cells are able to fight off pathogens of various types, and we're going to talk about that as we proceed. So all the leukocytes do provide defense and immune functions either within the bloodstream or by leaving the bloodstream and going into body tissues. The process by which they leave the blood vessel and move throughout the tissues or migrate in the tissues is called extravasation. And so in the blood here, we have a white blood cell watching other blood cells extravasate out of the blood vessel. And in the cartoon, you see that this white blood cell is saying, just follow the chemokines. Well, chemokines are just molecules that attract white blood cells to a certain site. And so the white blood cell is attracted along a gradient of these chemokines. And they squish in between the endothelial cells, the simple squamous cells, of the blood vessel and get out into the tissues. Now, the route that they take to the tissues will also depend on what tissue you're in and what chemical is being released by the damaged tissue. So this is a little bit more of a detailed synopsis of how extravasation occurs, not a cartoon version. We have damage or infection of tissue out here. Bacteria are present and probably damaging some body tissue. That tissue releases certain cytokines, or you could call it a chemokine. And the endothelial cells, in response, the blood vessel cells, make certain molecules called cell adhesion molecules, or CAMs for short. The CAMs on this picture are pink, various colors of pink. Those CAMs attract the neutrophils. So and other white blood cells, but neutrophils usually respond first, and the neutrophils bind to the cell adhesion molecules on the surface of the blood vessel interiorly, and they roll along the blood vessel, eventually finding a spot that they can squeeze through. And when they squeeze through the endothelial cells, that process is actually called diapedesis. And by an amoeboid-like movement, they leave the blood vessel and are attracted to the damaged tissue. So a little bit more information, the cell adhesion molecules in particular. Okay, let's look at the different types of granulocytes and what kind of protection they afford us. I'll start with neutrophils because they're the most common. They have a lobed nucleus, meaning it is all one nucleus, but you see enlarged regions and then narrow regions. So we call that lobed. And they fight bacteria predominantly. They can perform phagocytosis, which means, eating up the bacteria, engulfing the bacteria. They also can release antimicrobial or antibacterial chemicals, and we'll look a little bit more at that later. Eosinophils, on the other hand, have specific granules that contain antiparasitic enzymes. So parasites versus bacteria are fought. So eosinophils fight against parasitic infections. They also mediate some allergies. They are able to perform phagocytosis, but not of a whole bacteria, instead just of complexes of antigens, which are foreign molecules, bound to antibodies. And those antibodies are made by lymphocytes. And they attach to the antigen, and then phagocytosis by an eosinophil occurs. When we discuss the immune system, we'll talk about that in a lot more detail. For right now, you just need to know that eosinophils fight parasitic infections because they can release antiparasitic enzymes. Now, if we look at basophils, these cells are really unique because you can't see a nucleus. It's there, but you can't see a nucleus because it's so packed full of dark staining granules, specific granules. And these specific granules contain two compounds, histamine and heparin. Both of these increase blood flow. So a histamine causes a blood vessel to dilate, become wider. And so more blood will go to that particular tissue there. And heparin is an anti-clotting agent. So that will help blood flow without forming clots. So basophils we see in allergic reactions as well as tissue damage and infection. Okay, let's look now at agranulocytes, starting with monocytes. So monocytes fight viral infections. They actually transform into cells called macrophages when they leave a blood vessel. So in the blood, we call these monocytes, but when they leave the blood, we call them macrophages. So monocytes are noticeable because their nucleus is basically rounded, and it usually has this little indentation. The nucleus almost looks like a kidney, kind of kidney shaped. These monocytes can perform phagocytosis of virally infected cells. They also can convert heme to bilirubin, which we talked about before, and they can activate lymphocytes during an immune response. When they do phagocytosis of an infected cell, and so the antigen is now inside the cell, the foreign particles, the infectious agent, they have the ability to present that antigen out on the surface of the cell membrane. They process it and they display it. This is really important in an immune response, and so we'll talk about that again when we do the immune system. For now, you can just know that monocytes fight viral infections, and they can leave the bloodstream, go out to tissues. There they're called macrophages, and now they can do lots of phagocytosis out in the tissues. Okay, the other type of agranulocyte is a lymphocyte. These fight diverse infections, protect against cancer, and provide memory in our immune system. Before we get into the details of lymphocytes, let's first look at the structure. In lab, you're supposed to be able to identify the different types of white blood cells, so I want to mention that here. Notice that the nucleus is large and it's very round. And the biggest feature, I think, is that the nucleus takes up almost the entire cell diameter. Okay, so there's very little cytoplasm in all types of lymphocytes, which is what I'm going to talk about next. So there's three types of lymphocytes: natural killer lymphocytes, T lymphocytes, and B lymphocytes. Natural killer cells travel around the bloodstream. They can also get into the lymphatic vessels and take up residence in a lymph node. They provide immune surveillance because what they can do is destroy any abnormal cells. Any cells that have been infected with a foreign substance, any foreign cells that got in, virally infected cells, and cancerous cells. So they actually kill. Okay, they can do some damage, which is good, to an infected cell. Then there's T lymphocytes. These cells are referred to as T cells because they mature in the thymus. They're really good at coordinating the actions of a lot of different immune cells. So we're going to talk about their function in the immune system. They release lots of different chemicals that activate other leukocytes and immune cells. B lymphocytes are called B cells because they mature in the bone marrow. These are the cells that release antibodies against foreign antigens that have been introduced. So when you're infected with something, a B lymphocyte would release an antibody that is specific against that particular foreign particle. Okay, if we just look at the clinical data for like leukocyte counts, we see that we can that there's a normal number of each of these types. This type of lab test that's done is called a complete blood count with differential. And what differential means is it goes beyond just a total white blood cell count, the normal range of 5,000 to 10,000 cells per microliters, and actually reports on how many of each type of cell they saw. So normally neutrophils are the most prevalent, making up 60 to 70% of all the white blood cells. And the rarest are basophils, making up less than 0.5%. Please don't memorize these numbers of cells per microliter. Instead, I think just knowing the prevalence in percentages is probably adequate for our purposes. So neutrophils are the most common, lymphocytes are next, monocytes and eosinophils, and then basophils. I think there's a moniker to remember this: never let monkeys eat bananas. So that might help you. I don't know. Somebody came up with that, not me. Okay, let's look at some leukocyte disorders, just a couple. One is leukopenia, which means a low white blood cell count, and the other is leukocytosis, which is a high white blood cell count. What are some causes of low white blood cell count? Well, heavy metal poisoning can destroy white blood cells. Radiation can cause white blood cells to die. Certain viral infections will destroy certain subtypes of white blood cells. And there are some drugs that also affect your white blood cell count. Cortisol, glucocorticoids can. And then, of course, some people take immunosuppressants if they have a organ transplant. And certain chemotherapy agents will destroy white blood cells as well. And so the problem with leukopenia, of course, is you have less of an ability to fight infection. Leukocytosis, on the other hand, a high white blood cell count, is usually caused by the presence of an infection, sometimes due to allergy, sometimes due to dehydration, but that just means the plasma level is down, the water level is down, you know. So that just kind of amplifies the proportion of cells in a volume of blood. Anesthesia, surgery, and some cancers also increase a white blood cell count. So when you have damage in your body, white blood cell counts go up. But let's look at cancer in particular, leukemia. Okay. There's different types of leukemia. Myeloid leukemia increases the number of granulocytes and monocytes, whereas lymphoid leukemia increases the number of lymphocytes. You don't have to necessarily memorize that. We will look at slides in lab, and there's pictures in your lab manual of different types of leukemia. What you notice, though, is just a plethora of white blood cells. There's just so many more than usual. Now, the complications of leukemia are that the proliferation of all these white blood cells through the process of mitosis will replace normal bone marrow. So you'll get deficiency in other blood cells, like fewer red blood cells, fewer platelets, and maybe deficiency in other white blood cell types, depending on how specific the leukemia is. The other thing is that these cells are typically cancerous. and so they are either immature or defective. This leads to opportunistic infections. So if you're deficient in normal white blood cells, you can't fight infection. And if you're deficient in certain types of white blood cells, there are certain kinds of infection that you can't fight against. Okay, there's one other disorder that I thought I would mention, and that's mononucleosis. So mononucleosis makes you think of monocytes, right? But actually in mononucleosis, there's an increased number of lymphocytes. And it's due to the presence of a virus, Epstein-Barr virus. It can occur with herpes, but usually it's the Epstein-Barr virus. And the symptoms of the presence of this infection include fatigue, fever, swollen lymph nodes. If it's really severe, your spleen and liver become enlarged. And the virus itself is transmitted through saliva. It can be transmitted through blood or semen, but that's not as common. It's usually saliva, which is why it's called the kissing disease. Diagnosis of mononucleosis is confirmed not by the increased number of lymphocytes, but instead by the presence of antibodies that are specific against that virus. So that's our last disorder for leukocytes and the end of the leukocyte lecture. Thank you for listening.